everyone. My name is Jessica VASU and I'm from Polytechnic Nilai Negeri Sembilan. My matrix number is 23DBT 20F 2019 and I'm from DBT 3A. I'm now going to talk about the protein isolation by column chromatography. What is protein isolation? Protein purification, or also known as protein isolation, is a series of processes intended to isolate one or a few proteins from a complex mixture, usually cells, tissues, or whole organisms. Protein isolates are a good functional property such as emulsifying capacity, filmogenic properties, and water solubility.
Okay, now we are going to start the simulation. First, spin the EC tube in the microcentrifuge for 10 minutes to separate cells from the growth medium. We are going to place this balancing tube right in the opposite of the EC tube so that the centrifuge is balanced. So centrifuge look balanced and ready. Now we are going to close and set the timer for 10 minutes. So now you are going to start and wait until the timer runs out. Now we are going to remove both the EC tube and the balancing tube. Okay. So open the EC1 tube. So this is the EC1 and we are opening it. So open the waste collection jar. Okay, next. Tilt your tube over the waste collection jar to dump out the supernatant without disturbing the cell pellet at the bottom. So now we are going to pick the EC tube and just place it over the waste collection jar. Alright, now we're going to put it back and close it. Next, set the P1000 to 1000. So we are going to take this. Okay. So we are going to set this micropipette to 100 and save volume. Now we are going to open this tip and attach a tip to it. Okay. Now we are going to open the EC2 tube. To increase the amount of red fluorescent protein, we add more cells from the second tube. So we are going to open the EC2 tube and we are going to draw 100 microliters of liquid culture from EC tube. Alright. Now we are going to dispense the culture into the EC1 tube. Okay, there you go. Alright, now we are going to open the biohazard waste container and eject the tip into the biohazard waste container. Okay. Okay, we will place this micropipette at where it belongs and close all this. Now we are going to spin EC1 tube for 10 minutes. So we open the microcentrifuge, we place EC1 tube into this and the balancing tube. Okay, and we are going to place this balancing tube right on the opposite of EC1 tube and check the balancing. Okay, centrifuge look balanced and ready. Now we are going to close and set the timer for 10 minutes. Start. Okay, now we are going to close and remove the EC tube from the microcentrifuge. Okay, close and open EC1 tube. Open the waste jar container. Okay, it's already opened. And tilt your tube over the waste jar container to dump out the supernatant without disturbing the cell pellet at the bottom. Okay. Alright. I'm going to close this and close this. Okay, next. Okay. Set the P200 to 150 microliter. So we are taking the P200 micropipette and set it to 150. Alright, save volume, and we are going to attach the tip. Okay. Now we are going to open the EB tube. This is the EB tube, and draw up 150 microliters of elution buffer, which is the EB. Okay. Now we are going to dispense the culture into the EC1 tube. Alright, so we are going to eject the tip into the biohazard waste container. We are going to eject the tip and place it at its place and close this. So next, vortex the EC1 tube for 10 seconds to resuspend the cell pellet. So we are going to take this EC1 and place it over the vortex and put it back in its station. Attach a tip to the P200. Okay, P200. Okay, open the LYB tube. So lysis buffer and draw up 150 microliters of lysis buffer. And dispense the lysis buffer into the EC1 tube. So we are going to close this tube and open EC1 tube and dispense it into this. Okay. Now we are going to open the biohazard waste container and eject the tip into it. Check. Okay. We are going to set a timer for 12 hours to incubate EC1 tube overnight at room temperature. So timer. 12 hours, start. Sorry, there occurred a mistake. I forgot to centrifuge the tube. Now we are going to centrifuge the tube. Now we are going to take out the EC tube from the microcentrifuge. And we are now going to set a timer for 12 hours to incubate. Okay, now next, place the waste collection jar under the valve of the chromatography column. So we are going to take this and place under here. Open the column to drain away the liquid. Okay, done. Now spin down the EC1 tube for 10 minutes and then remove the tubes from the microcentrifuge. Balancing tube. Okay, balancing. 10 minutes. Now we are going to remove the EC tube from the microcentrifuge. Okay, close. Now set the P200 micropipette to 200 microliters. So we are going to take this 200 and set it into 200. Okay, save volume and attach a tip. Okay. Now open the EC1 tube and draw up 200 microliters of supernatant. 200. Now we are going to close this. Open the super tube. We are going to open the super tube and dispense the supernatant into the super tube. Supernatant into the super tube. Okay. Alright. Now we are going to close this and open the biohazard waste and eject the tip into the biohazard waste container. We're going to place this here, close this. Okay, next, attach a new tip. I'm going to take this and attach a new tip to it and open the BB tube. So this is the blinding buffer, and draw up 200 microliters of the binding buffer. Now dispense the BB into super tube. So we are going to open this and dispense it into here. Okay. Now we are going to open the biohazard waste and eject the tip into the biohazard waste container. So here, eject tip. Alright. And close this. Alright. Next, pick up the P1000 micropipette. Okay, P1000, and open the tip box, attach a tip, and draw up 400 microliters of the super solution. So we are going to set this to 400. Okay, save volume. So we are going to open the super solution tube and draw 400. Now dispense the red super solution into the chromatography column. So we are going to bring this over here and dispense it. Okay. Now open the biohazard waste container and eject the tip into the biohazard waste container. Alright, eject. I'm gonna keep this here, close this and close this super tube. Place the waste container jar back under the valve. So, okay. Open the column and allow the liquid to drain into the waste container. Close the valve when the liquid is just above the resin bed. Alright. Okay, done. And we're going to place this back here. Okay. Now next, set the P1000 micropipette to 1000 microliters and attach a new tip. Save volume, and we are going to attach a new tip to it. Okay, close. Dispense 1000 microliter of the WB solution into the chromatography column. So this is the wash buffer, which is also known as WB, and we're going to dispense 1000 microliters of it. We're going to take this over here and now we are going to open the biohazard waste container and eject the tip into the biohazard waste container. Eject. Place it back. Now we are going to open the column and allow the liquid to drain into the waste container and close the valve when the liquid is just above the resin bed. Okay, now we are going to and done. Okay, next, using a new tip, draw up 1000 microliters of the EB solution. Dispense gently down the side of the column. Repeat this step. Okay, So we are going to take the EB, which is the Edophant buffer. First we are going to take this P1000 and attach a new tip to it, and we are going to draw 1000 microliters of the EB solution, and down the side of the column. Okay, alright, now we're going to place this now we're going to open the biohazard waste and eject the tip first. Okay, now open the column by turning the valve. Watch the red protein traveling through the column. When it nears the bottom, close the valve. Okay. Okay, done. Now place the open RFP tube under the valve. So we're going to take this out and open this RFP and put it under the valve. And now open the column to capture the RFP. Alright. Open. Alright. Close the valve and RFP tube. Close it. Done. Done. Okay. Put it back here. Now place the waste collection jar back under the valve. Alright. Now dispense 1000 microliters of the column equilibration buffer CEB solution into the chromatography column. So this is the column equilibration buffer. We're going to attach a tip into this and dispense 1000 microliters of the solution. And into the chromatography column. Now we are going to take this out and eject the tip. Eject. Place it here. Close. Now open the column and allow the liquid to drain into the waste container. Close the valve when the liquid is just above the resin bed. So we are going to open this valve. Open. Okay, done. Repeat 3 times before storing the column away. So we are going to take this solution another one time, attach the tip and go take it. Okay. Place it in here. Check. Once. Okay, another one time. twice and another one time. Right. Okay, ejecting. Place this here. Close this. Close. And the tries for the third time. Done. Okay, next. So this is the result. This is the ideal cell culture result. Red, slightly cloudy, because the bacteria replicated in LB, AMP or ERA are produced RFP, which remained inside the cell. Predicted result. Actual results. And ideal results. Okay, thanks for watching. That's all from me. Bye bye.